Hello dear learner, in this session I will take you through a types of CRISPR-Cas and its a mechanism in brief by which a bacteria defends against bacteriophage. At the end of this session, a learner will be able to brief about the types of CRISPR-Cas in bacteria, a defense mechanism and a significance of CRISPR-Cas in bacteria. A CRISPR-Cas system can overall be classified into class 1 and class 2, and which in turn, depending upon a types of Cas protein, divided into 6 types. Of course, a type of Cas protein is not the only criteria to put into various types. A class 1, which includes type 1, type 3 and type 4, whereas a type 2 covers up type 2, type 5 and type 6. Class 1 is characterized by the presence of a complex of several small Cas proteins, whereas a class 2 is characterized by the presence of a single large Cas protein for the degradation of foreign DNA. The 6 types are further subdivided into 19 subtypes depending upon a Cas variant protein. A Cas protein which always forms a cluster of genes and present always next to CRISPR locus in a bacterial genome. Overall 93 Cas genes have been identified so far, which are further grouped into 35 families depending upon the sequence similarities. In this adjacent table, one can see 10 core Cas proteins numbered from Cas1 to Cas10. A Cas3, Cas9 and Cas10 are referred as signature proteins because they are the one which eventually guides a Cas protein for the degradation of the target DNA. Whereas Cas1 and Cas2 are ubiquitous protein and helps a bacteria in acquiring a spacer sequence from a mobile element or an invading bacteriophage, whereas other proteins such as Cas5, 6, 7 and many are known to help in a maturation of CRISPR RNA during a defense mechanism against a bacteriophage. It is also established by biologists that in a single bacteria or a bacterium, a more than one type can coexist. When a bacteriophage enters inside the bacteria, how do bacteria tackles this bacteriophage and inactivates this infection? This overall process can be divided into 3 stages depending upon the set of events. A first stage is adaptation or acquisition. When a bacteriophage infects a bacteria may acquire a part of a DNA sequence that we call it as spacer, as we have learnt in session 1. Whereas in a stage 2, a CRISPR locus undergoes a transcription to form a precursor CRISPR RNA, which is further matured by a set of Cas proteins to form a mature CRISPR RNA, which then associate with Cas protein to form an active CRISPR-Cas complex. Whereas in an ultimate stage, it is this CRISPR-Cas which in a sequence-specific manner brings about a degradation of a foreign DNA or a bacteriophage DNA and thus leads to abandonment of a bacteriophage life cycle and thus bacteria wins a battle over a bacteriophage. A CRISPR-Cas is a highly specific sequence, all because of RNA. As the sequence or as this endonuclease protein or a Cas protein is guided by RNA molecule at a target sequence. However, a very key step which is responsible for this specificity of CRISPR-Cas, very less knowledge have been available so far. However, it is known that there are two types of spacer acquisition, one is naive and one is prime. And it is Cas1 and Cas2 complex which helps bacteria in acquiring a spacer from a bacteriophage. A naive spacer acquisition is a spacer acquisition whereby when a bacteria acquires a spacer sequence from a bacteriophage from the very first time, and therefore it has now become an immune to bacteriophage and subsequent infection cycles. Whereas a prime spacer acquisition is an acquisition whereby a bacteria is already immune to a bacteriophage. However, this time bacteria acquires an additional or a new spacer sequence from the same bacteriophage. A type 1, which is also known as a CRISPR-Cas3 because of a signature protein Cas3. A type 1 system is characterized by a complex known as cascade complex, which is a giant complex made up of several subunits of proteins, as it can be seen in this adjacent diagram. Upon expression of CRISPR locus and formation of precursor CRISPR RNA, it is processed by a Cas5 or Cas6 protein to form a mature CRISPR RNA, which then associate with cascade protein complex, which in turn recruits Cas3. Cas3 is an endonuclease enzyme, which in turn brings about degradation of a foreign DNA or bacteriophage. A type 2 CRISPR-Cas, which is also known as CRISPR-Cas9 system, which is most widely used by genetic engineers as a tool for genome editing, just because a Cas9 is encoded by a single gene, whereas in other system the cascade or the other similar complexes is the result of several small genes and their expression forming a giant complex. Apart from its role in degrading target DNA, Cas9 are believed to play a role in acquisition as well as a processing of CRISPR RNA to form a mature CRISPR RNA. Upon expression of CRISPR locus, when a precursor CRISPR RNA is formed, it is processed by an unknown factor, RNase 3 enzyme, and a Cas9 enzyme to form a mature CRISPR RNA, which then associates with Cas9, which is also assisted by a tracer RNA. A tracer RNA which stands for transacting CRISPR RNA, which forms heteroduplex with CRISPR RNA as we have seen in earlier session. A tracer RNA are believed to play a two key role. One, it helps in maturation of CRISPR RNA, and second, after forming an heteroduplex with CRISPR RNA, it guides Cas9 where to introduce a double-stranded break in the DNA. A Cas9 introduces a nick next to PAM within the protospacer sequence. A CRISPR-Cas9 sequence is an PAM-dependent sequence. It simply means that a Cas9 scans for the presence of a PAM from a bacteriophage sequence. And this is the reason why bacteria does not cleave its own genome, though it has a spacer sequence which is equivalent to a protospacer sequence, just because a bacteria lacks the presence of PAM sequence. So in a very first stage, a CRISPR-Cas9 scans for the presence of PAM. Once PAMs are recognized, a CRISPR RNA forms an heteroduplex with a protospacer, precisely a spacer sequence from a CRISPR RNA which forms a heteroduplex or a hybrid with a protospacer sequence from a bacteriophage, which in turn leads to an activation of CRISPR-Cas9, and that leads to introduction of double-stranded break or a break in the double-stranded DNA, or DNA is fragmented, and thus a virus life cycle is abandoned or terminated because a fragmented DNA with which a virus cannot complete its life cycle. It is also identified that the spam sequences are specific for a specific Cas9. For example, a, a Cas9 from most widely used Streptococcus pyogen, in short SPCas9, the respective PAM sequence is NGG. A PAM sequence which is mostly a requirement of several CRISPR-Cas, however not of type 3. This slide sums up a specific sequences for a particular Cas9 protein. A type 3, which is also known as CRISPR-Cas10, as it is characterized by the presence of Cas10. A type 3 system has a cascade equivalent complex known as CSM or CRM complex, depending upon a subtype of type 3 system. Though a Cas10 is a signature protein of type 3 system, however, its precise role associated with type 3 is still not so very clear. Upon expression of a CRISPR RNA, it is matured by Cas6, an unknown factor, to form a mature CRISPR RNA, which in turn associates with CSM or CMR depending upon a subtype of type 3, which in turn recruits Cas10 and degrades DNA as well as RNA. A point to be noted over here, unlike other systems which can only degrade DNA, a type 3 system can also degrade RNA. However, who brings about a degradation of this foreign nucleic acid? It's still not sure whether it is Cas10 or whether it's a component from CSM or CMR. However, many of the literature are in favor of Cas10 performing this task for the type 3 system. This slide sums up some of the key features of type 1, type 2 and type 3 as discussed in earlier slides. So as established in the year 2007, a CRISPR-Cas system helps bacteria becoming immune to a particular bacteriophage, and therefore a subsequent infection, that particular bacteriophage is inactivated or the DNA is degraded in a very specific manner without breaking its own genome. At the same time, it is this immunity which is acquired by a parent bacteria can be passed on to subsequent daughter cells or progenies as well. Though this role is very key and have been identified so far as the only role, biologists are looking for a role of CRISPR-Cas in the direction of repair or gene regulation, or is it really associated with evolution of bacteria in any way? Whereas genetic engineers are busy exploiting this fundamental system for bacteria for the purpose of genome editing, whereas viruses are busy involving a mechanism by which they can become resistant to this CRISPR-Cas system. And so far, two key mechanisms identified are: one, by simply introducing a mutation within a PAM, or by introducing mutation or some kind of alteration in protospacer, so that a bacteriophage sequences cannot be identified by a CRISPR-Cas system. Thank you.